Hello, and welcome to our discussion of integrated photonic biosensing. I'm Ben Miller. I'm a faculty member at the University of Rochester Medical Center, and today I'm going to take you on a fairly rapid tour through some of the ways that we can develop integrated photonic biosensors and what we can do with them. So the first question is, why do we want to work with sensors? Well, for those of you that hear about big data and think about all of the things that we can do with large amounts of data and information, all of that data has to come from somewhere. And if we're interested in our world, whether we're talking about the globe as a whole or ourselves as individual organisms, that data has to be derived from the analog world via some kind of sensor. So a sensor is the interface between the universe and data. Thinking about this on a little bit more prosaic scale, sensors really give us the opportunity to develop all sorts of applications ranging from the home to clinical laboratories and research laboratories and everything in between. Photonic sensors have particular advantages of size, weight, and power. Just like the advantages that integrated photonics has for other applications, those pertain as well to biological sensing with integrated photonic sensors. But there's also more in that one has the opportunity to combine multiple sensing modalities. I'll talk a little bit about what that means later on. But also, because we can design structures that have very small mode overlaps with the photonic device, we can make systems that are extraordinarily sensitive, much more so than we could do with any other kind of macroscale sensor. So another thing that's particularly important is that integrated photonic sensors give us the ability to have individual elements of the sensor that all operate independently, able to detect different things that we're interested in. So one analogy is for those of you that might have had a Lite-Brite as children. In a Lite-Brite, each one of these little pegs is a reporter element for light, and together they make up a picture that you might have put together using different colors and so on. But with integrated photonic sensors, each one of these reporters could be a responding element of a photonic sensor, can be a different biological target that we want to detect. So, for example, a network of proteins in the human body, and we can use that information to tell us about our own health and about pathogens that might be attacking us, things like that. So that's the kind of thing that we'll talk about in our discussion today.